Okay, so we're looking at Roman numeral 4 in your outline, macroevolution. So macroevolution is the evolution of organisms above the species level. So we're not talking about making a new species now. We're talking about making a new family or a new class or a new order, right? So in that taxonomic grouping, you're making something really, really different. And that involves sometimes making completely new body plans. So you can see how does a fish with no legs, how does something like that evolve into something that does have legs, right? So one of the things that we need to think about is just the time that is involved in the evolution of life. So this is just a picture of geological time or geologic time. And from the very first cell over lots and lots of time to where we get the different animals and plants that we have on the planet now, it's been, millions and millions of years. And so it's hard to think about that. It's a little bit mind-blowing to think about a million years, let alone 486 million years, and what kind of drastic changes can happen to living things. But we can look at dog breeders that breed dogs, right? So even if you had two very different dogs, like a Chihuahua and a German Shepherd, and you bred them together, right, the offspring would look very, very different. So you can sort of see in something like that where you can get a very different looking offspring in just one generation. So think about millions of years. And so let's take a look at the geological time and fossil record. So we know that because of the fossils and their order in the dirt, we can look at number one and say, like, yes, we can tell the relative age of fossils. It's the order in which they evolved. But there's no absolute number on the age of a fossil. So this is going to just give you an example of how you can put an absolute age on a fossil. It's called carbon dating or radiometric dating. And it's the most common method for dating our fossils today. And so let's understand the concept here. So there are three isotopes of carbon. So I'm talking about carbon dating. Three isotopes of carbon. There's carbon 14, carbon 12, carbon 13, and carbon 12. Remember when we talked about this in like the very first few weeks of class, you learned what is an isotope. An isotope is the same atom, but just different weights, right? They have different numbers of neutrons. So carbon 14 has 2 more neutrons than carbon 12. And over time, what happens is carbon 14 turns into carbon 12. So what I'm going to do is look at this example. So in a living organism, in this case, they're looking at a clam. Living organism is taking in from the environment when it eats, right, the carbon in its food. So carbon 14 is taken up by the clam in trace quantities along with much larger quantities of carbon 12, right? So carbon 12 is basically the form of carbon that's most prevalent in the atmosphere. Carbon 14 is more rare, but carbon 12 and 14 and 13 for that amount is all coming into that clam. However, when the clam dies, the clam stops eating, right? So it doesn't take in any more carbon. So the amount of carbon 14 that it has in its body will begin to decay. And that decay is called radioactive decay. So when it decays, it actually becomes carbon 12. Then you can look at, when you find a fossil, you can look at how much carbon 14 is left in that fossil. And you can determine how much time has lapsed because scientists know how long it takes for carbon 14 to become carbon 12. And we also know how much carbon 14 is in the atmosphere. And then how much, if you know how much you're starting off with, if you know initially like the ratio of carbon 14 to carbon 12 in the living clam, and then you find half of that in the dead clam, you know that time has, enough time has passed where half of that has decayed. So let's look at an actual problem and hopefully this will make more sense. Okay. So the way that radioactive decay works is in half-lives. So at time 0, okay, you're going to have 100% of the amount of carbon-14 in a living organism, right? Because you're actively taking in carbon-14, and let's say you just die, you still have 100% of that carbon-14 in your body. But over time, that is start to radioactively decay, and they measure decay again in half-lives, and we know that it takes 5.6 thousand years for the half of that carbon-14 to become carbon-12. Okay, so after 5.6 thousand years, we're going to have half as much carbon-14 in our body than we should, right? Then what the environment dictates. If we're still eating and we're still taking in carbon from our environment, we would still be up here at 100%. But since we died and we're not eating anymore and the carbon-14 is just sitting there in our bodies, right, it's going to start to decay at this rate that we know. So the half-life is 5.6 thousand years. So we can expect, if our bodies were exhumed 5.6 thousand years from now, half of the carbon-14 would be still in our bodies, but half would have decayed. Okay, so what about if another half-life happens, right? So 5.6 plus 5.6 is 11.2, so that's 2 half-lives. That means that half of this amount, right, is going to be in that fossil. So that means it's going to be 25%, right? Because half of 50 will be 25%. So if you find a fossil and you dig it up and you find that it has 25% of the carbon-14 it should have, then you can say, oh, well, if it only has 25%, that means 2 half-lives have passed. That means that we can put the date of death at around 11.2 thousand years ago. Okay, and it continues on that all the time. So another half-life will be 16.8 thousand years, and you would find half of 25, so something around 12.5% of the carbon-14 in that fossil, right? And on and on and on. So you basically, when you find a fossil, you look at how much carbon-14 is in that fossil, and let's say it is something like 10% of what you would normally see in a living thing. Well, then you can say the life of that fossil is somewhere around here, right? Because this graph is at 12.5%, right? So you can say that that fossil is roughly 16.8 thousand years old, okay? So let's look at a problem here. So a paleontologist estimates that when a rock formed, it contained 12 milligrams of radioactive isotope potassium-40. So don't worry, we're using potassium-40, but it's the same concept. The rock now has 3 milligrams, so it went from 12 to 3. The half-life of potassium-40 is 1.3 billion years. How old is the rock, right? So let's see, how many half-lives has happened from 12 to 3? So what's half of 12 is 6, right? So that's 1 half-life. And then half of 6 is 3. So that's 2 half-lives passed to get to 3 milligrams. So we know 2 half-lives have passed. Each half-life is 1.3 billion years. And so we just 1.3 plus 1.3 is going to give us 2.6. So this is your answer. The rock is 2.6 billion years old. Okay, so if you get a question like this in your test, which you are most likely going to, you have to just look at how much of something, whatever you're looking at, whatever mineral you're looking at, look at what it started off with, look at what it has at the end, calculate how many half-lives has passed, and then just add your half-lives up, okay? And then you got how old something is. All right, so that is radiometric dating. The next topic here is going to be homeotic genes, large effects from small genetic changes. So at this point, I think we've been talking about genes in terms of like one gene makes one protein and it has one small effect on the body. But in reality, hold on just a second. Okay, I just wanted to fix the spacing here. All right, so in reality, sometimes there are sets of genes in our body that really are responsible for making very large body plan alignments. The structure of our body, like we have a head, our head goes up here, we have four limbs, the two arms go here, the two legs go there, so those genes are called homeotic genes. Homeotic genes are genes that shape your body plan. So in the evolution of invertebrates to vertebrates, it turned out that all that happened was that the fruit fly, which is an invertebrate, has a single cluster of homeotic genes. But mice have four clusters of homeotic genes. Two clusters, right, seem to have occurred in the evolution of vertebrates. Mutations in the duplicated genes may have led to developing the jaw, the backbone, and limbs, right? So these bones and limbs that we find in our mammals and different animals, our vertebrates, exist because of these different, we have more clusters of homeotic genes, and they are actually expressed more than once, whereas these guys have a single cluster of homeotic genes that are expressed once. So the expression pattern of homeotic genes can really dictate what your body looks like. And they've done lots of experiments with these homeotic genes, mutated them, seen what they do when they get mutated, and you get really wild results. So that is an example of how you can get very different body plans from homeotic genes. Okay, here's a different look at homeotic genes controlling the body plans. So the color coding refers to just the regions of the body that are similar in terms of the genetics. And we can see that the different kinds of body plans here can be due to the control of different homeotic genes. And then this is another example of how could feet evolve from fins. There are one or two expressions of homeotic genes. If you get one expression of homeotic genes, they develop into fins. The second expression of your homeotic genes will create skeletal features of limbs. So to go from a fin to a limb is just one more round of homeotic expression. So hopefully this is making a little bit of sense to you, right? That the concept is that you have these genes that can really change body plans drastically. And so even though we talk about evolution and body plans as this, long change over time, it is, but this is actually something that can happen very relatively quickly. Okay, our next concept here is adaptations of old structures for new functions, which is called an exaptation. So what it is basically you have something old, there's something that exists in nature, and it's just used for something else, and it became successful as being used as that thing. So feathers is one of the most prevalent examples of an exaptation. So remember in our last lecture, I talked about how our dinosaurs had feathers, and there was a fossil that looked at that saw this, and so we discovered that some of our reptiles had made feathers, and this is an example of that. So these are the different stages of feather development. Okay, so initially the feathers were believed to have evolved from scales, right? Scales that exist on our reptiles. And this keratin is actually spelled wrong. I ripped this off from the internet, but anyways, the protein here is the same for your scales and also for your feathers. And what the feathers were thought to be used for initially is for insulation, to keep the reptiles warm, and they weren't really used for flight. But it turns out that those reptiles that had feathers were better at flying, right? So then the feathers were over time were more and more, developed to enhance flight. And now we have these very intricate feathers today that birds use for flying. Okay, so you don't have to memorize any of these different steps or anything. It's just a nice example of exaptations. All right, and then our last example here is moving from a simple structure to a more complex structure. So I'm going to post this video of Neil deGrasse Tyson going over the evolution of the eye, and I'm going to create a little quiz because I want you guys to watch it, and it'll be part of your lab assignment. So you'll see that in your Canvas. But what you look for is, or what we're talking about here is there are very, very simple eyes that exist in the world where you have a few light sensitive cells that are found in organisms that still exist today. And you also have very complex eyes, like the human eye, for example, right? Or in this case, the squid eye, where you have the development of a lens and you have different areas and you have a fully formed retina and different layers of the eye. So this is an example of a large change, right? So you have, but it really came from a simple structure in the past. And so given geologic time, given the amount of time required for things to really become formed and to become sort of utilized and passed on and functional, the eye is a good example of something like this. So again, I'm going to post this so you guys can take a look at that and answer the questions to that. Okay, and that is it for this chapter 14.